start. Welcome back after the break session. This is our last session for the first day of the conference. Before the closing remarks and the networking activity, we are going to have two more presentations. The first presentation is from Mariona Pinart from Max Delbrük Center for Molecular Medicine in Germany. Mariona is going to present us her results from a DataSHIELD analysis nutritional epidemiology study within the ENPADASI project. Mariona, can you share your screen, please?
I'll try. Let's see. Share. Let me see. Okay, so I'll go. I have my presentation here. Okay. Okay, so can you see my screen?
Yes.
Brilliant. So as you very well said, I'm going to present some of the results from an already published paper. Actually, it was published in February this year. So we published federated meta-analysis using DataSHIELD in the field of nutrition epidemiology and within ENPADASI initiative. So basically, ENPADASI was a consortium, and as you can see, this initiative is long over, but we're basically a consortium of around 50 partners from like 9 different countries across Europe. So it was a large consortium, involved in a very short term project. We had we delivered an open access research infrastructure with the aim to share data and query metadata from a wide variety of nutritional studies identified within our consortium, that covered intervention and observational epidemiological studies, with the ultimate goal to conduct research analysis and to conduct a combined analysis. Said and done, we queried metadata from the observational studies, and we decided that we were in the position to conduct a research project, and it was to investigate the relationships between a dietary macronutrient composition and the lipoprotein profile in European adults. This is a cross-sectional study, and we decided to use DataSHIELD. And the reason why we use DataSHIELD, I think it's like everybody, it's a common reason, is that the owners of our of observational studies were very keen on being involved in research projects, but they were quite reluctant to share the data. So to put you a little bit into perspective, why did we choose this research topic? It's because, as you know, cardiovascular diseases is the most common cause of death worldwide. Right, it's accounting for over 4 million deaths. That's a public health problem. Furthermore, 50% of the cardiovascular diseases are attributed to alterations in the lipoprotein profile. Actually, European guidelines recommend that as primary targets therapies that reduce, that lower the concentrations of total cholesterol and particularly LDL cholesterol for the prevention of cardiovascular diseases. However, it has emerged a new target for cardiovascular prevention, which is the non-HDL cholesterol. Actually, some meta-analysis have found that a non-HDL cholesterol correlates better with cardiovascular risk than the LDL cholesterol. And as you can see depicted in this picture, you see that here we have all the cholesterol, and basically the good cholesterol is the HDL, and then the rest is the atherogenic lipoprotein, so the bad cholesterol, which includes also the LDL. Basically, we know that diets that are low in saturated fatty acids have beneficial effects on the lipoprotein profile because it decreases the LDL cholesterol. Whereas, for instance, diets rich in carbohydrates, they have a detrimental effect on the lipoprotein profile because they reduce the good cholesterol, so the HDL, and they increase the triglycerides. All this evidence that have shown comes mainly from the intervention studies of small sample size, which precludes the assessment of differences between age, differences in age and sex. Moreover, there are not many, if any, studies looking at the associations of isocaloric replacement models and non-HDL cholesterol. So we took advantage of this, and we're thrilled to investigate the association of isocaloric replacement of carbohydrates with total fats or different type of fats with HDL cholesterol, which is the good one, and the non-HDL cholesterol, which is the bad one, in 8 European observational studies. Like I said before, I'm going to show you some of the results because we also stratified by age, sex, we looked at interactions, etc. But I'm going to focus on this main aim. So I'm going to go briefly to the methods. I'm going to take this out because I cannot see anything, sorry. Okay, good. So here we have the study population. Our study was not as big, as large as my colleagues, the ones that presented earlier. So we only have like about 6,000, nearly 6,000 participants, which was not bad, age 13 to 80 years old. There were mostly from Germany, I have to say, so 5 out of 8 studies were from Germany, and the other 3 were from Belgium, Italy, and Spain. As I said before, if not, I say it now, our federated analysis our case study is of cross-sectional design, and so we had 5 studies that were of cohort design. So we request data at baseline, either at baseline or at single follow-up. And as you can see, regarding the sample size, we had very small and large studies, like Active-E from Germany had only 50 participants, whereas the Gini plus Lisa, also German study, had with more than 2,000 participants. As you can see, our exposure, the data macronutrient composition, was assessed using different dietary assessment tools like the 24-hour recall, the food frequency questionnaire, the food records, and other tools. So this should be taken into account. Why? Hang on. Okay. It's stuck. So as exposures, we requested information from carbohydrate intakes, total fats intake, and then the saturated, monounsaturated, polyunsaturated fatty acids, and the protein intake, and as outcome variables, the lipoprotein profile, like mainly HDL cholesterol, LDL cholesterol, and total cholesterol. And as of variables, here I depicted a few actually. It's the age, sex, weight, height, smoking status, energy intake, so the total energy intake from fats, carbohydrates, protein, and alcohol, and also alcohol. Initially we asked for more covariables, but as one of the presenters said, not all the studies. So we also had the issue that not all the studies had the same, had the variables, because the variables, like for instance, we asked for waist circumference, and not all the studies had it. So we had to deal with, in order not to lose power. So obviously, as you all know, previous effort of harmonization effort should be done, because all the data sets have to have the same, they had to, all the variables have to be spelled the same way. It's very important, and using the same units, and etc. So in order to conduct the analysis from the client side. For this particular project, we had to create some new variables. So from height and weight, we created a BMI. We did it, using DataSHIELD functions. We also categorized some numeric variables, like for age and alcohol. We collapsed categories from a categorical variable like this smoking status, and the reason why we did that is because we had Ginny and Lisa with study that had teenagers, and it's I wouldn't say impossible, but it's very unlikely to have former smokers in a teenage population. We also created a variable of percentage of energy available from carbohydrates, proteins, and fats in order to conduct the isocaloric, the energy density models. And we also, very important, we calculated this biomarker, the non-HDL cholesterol, by subtracting HDL, so the good cholesterol, to the total cholesterol. And we also created the study source. Okay, so in order to estimate the relationships between isocaloric replacement as 5% of energy of carbohydrates with total fats, or the different type of fats, and blood circulating lipids, we conducted multivariable nutrient density models, basically general linear regression models. And we started with a very, initial version of a DataSHIELD, and we end up publishing using the DataSHIELD version 5.0. While I think that the version number 6 was developing, but we decided, because we didn't have this armadillo platform, we had to to stick to to this version, because as it has been said, all the servers, all the partners should have, all the institutions should have the same version. So we stopped at version 5, and so we conduct pooled analysis. So at individual level analysis, and study level meta-analysis. So we use the DS GLM and the DS GLM SLMA. And for the forest plots, we had to, basically we run the DS GLM LS. The GLIM model for study level meta-analysis, and we retrieved the beta coefficient and the standard error, and then we created an Excel file, and then used what I say, RStudio, normal R, and we used the package metaphor, I think, yes, and we created the plots, the forest plots. So the way to read the output, like the beta coefficients, that coefficients indicate differences in blood lipid concentrations associated with the replacement of 5% of energy intake from carbohydrate with the equivalent energetic amounts of dietary fats. And we ran 2 models. The first model was adjusted for percentage of energy from proteins. So basically we had the outcome and then the exposure of interest, which was like, for instance, total fat, and then it was adjusted for a percentage of energy from proteins, which means that the percentage of energy from carbohydrate was left out from the equation. So we adjusted for total energy intake, alcohol consumption, both the categorized variable and the continuous variable, and the study source. And we also conducted model 2, which was basically model 1, but further adjusted for age, sex, smoking status, and BMI. We kept the significant levels at the p-value below 0.05. And for variables with missing data, in our case were like smoking status and BMI, we went by successfully using the simple imputation. Multiple imputation at that time was not available, so I'm very much looking forward to the next presentation. And once we submitted the paper, one of the reviewers pointed to the assumptions. So he was wondering of whether some violations in the linear regression assumptions were violated, right? And so we looked at the assumptions of linearity, homoscedasticity, and normality, basically using visually. It was all made visual with histogram and scatter plots that were that could be shown using DataSHIELD, and we could calculate regression residuals and feeder values. I'll show you later how. So let's go to the results. So basically here I show you the baseline characteristics of the participants. I would like to draw your attention to one particular variable, which is the age. As you can see, we had teenagers, so the Legionellies had teenagers. Donald's study had young adults, age 18 and above, like around 20. And the rest of the studies had like similar ages, except for the EPIC sub-study, which had like, older people. And I also like to draw your attention with these 2 variables, the energy, the percentage of energy from carbohydrates and the percentage of energy from fats. And it's because, as you can see, the first one, the percentage of energy from carbohydrates, you see that except for Ginny and Lisa, the rest, the studies, they had a percentage lower than 50%, and according to the European nutritional guidelines, it is recommended to basically eat around 50 to 50% of carbohydrates, and also this is depending on the countries, but mainly many European nutrition societies limit the intake of fats down to below 30%. As you can see, we had all our studies were like fat consumers, right? So as a recap, we had participants that ate less carbohydrates than recommended and more fats than recommended. They should be taken into account too. I'm going to show you now the results of the pooled analysis. So the so-called individual level data. And I'm showing for total fats model 1 and model 2. And as you can see, So when carbohydrates were replaced with total fats, we see an increase, a significant increase in the good cholesterol and a non-significant decrease in the bad cholesterol. And here I'm gonna highlight model 2 because I would like to compare it with the forest plot from the study level meta-analysis. I know it's quite, well, it's difficult to see, but you have to believe me that results were very, very similar in the pooled and in the study level meta-analysis. And also the heterogeneity was very low, like 21% here and 10% here. And if we look at the models for the different type of fats, we see that when carbohydrates were substituted with saturated fatty acids, which are like well, not so healthy fatty acids, the non-HDL was increased. And so it shows that it had detrimental effects upon the lipoprotein profile, whereas the unsaturated fats had a positive, so a beneficial effect, because, for instance, but in a different way. Meaning that whereas monounsaturated fatty acids increased the good cholesterol, the polyunsaturated fatty acids decreased, but the concentration of the bad cholesterol. And if we compare it with the study level meta-analysis, we see that, well, as you can see, well, for the saturated fatty acids, it follows the same trend, pretty much the same, so it's the same direction, pretty much the same magnitude, but the significance was lost because in general, our study level meta-analysis, well, the 90% confidence intervals were wider, were broader compared to the individual, to the pooled analysis. However, we see that we have, the same results were similar magnitude for the unsaturated fatty acids. And with regard to the linear regression assumptions, we tested the multicollinearity. So basically, what we wanted is that all the independent variables should be really independent to each other, to one another. And so we use the DS correlation. You can do it one by one, but it's very tedious, like variable 1 and variable 2. And what we did was to create a database, a data frame, with only the numeric variables. And so we ran also the DS correlation, and we had this table. And there's a rule of 9, as you all know, that if they are like between minus 0.5 and 0.5, we can say that they are like fairly independent. However, we have to admit that for age and BMI, there was a bit of a dependence, but very largely. So we thought that this first assumption was not violated. For the linear relationships between the dependent and independent variables, we conducted general linear regression models. We added the quadratic terms for the percentage of fat and different types of fat together with the linear terms. And we saw that there was no poor fit. Well, we saw that the relationships were linear. We also checked for the homoscedasticity, that is, that the variance of residuals are kept constant, and that the residuals were independent to each other. I think that Paul mentioned it, that it was very difficult, but there are no ways to provide use tests, so everything had to be very visual. So with scatter plots like this one. And so to achieve this scatter plot, we've had to first run the regression model, with the DSGLM, for instance, and then you get the output, with the beta coefficients. And then we use the beta coefficients to create the fitted values using the function dsmake. This is a very important function, actually, dsmake. And then once we had the fitted values, we created the residual values from the fitted values using, again, the dsmake. And we in this case we wanted to use standardized residual values, and it was a bit tedious because we had to more or less manually do it, using these functions, the ds bar, and ds length, and create it again using the dsmake. And then finally with the ds scatter plot, we managed to have this beautiful graph in which you can see that this line is the mean of the residual should be close to 0 ideally, which they were, and the residual should be.
Sorry for the interruption.
Yes.
Can you, to have a few minutes of questions, can you finalize?
Oh yeah, sure. And then I'm done. So yeah, so I didn't know. And so the normal distribution with the histograms, everything was done non-disclosive. For more information, you have Dimitri's paper, and basically federated analysis. Yeah, so we had similar effects between pooled and study level meta-analysis. We got consistent results. And every study specific differences, like different dietary assessment instruments, or the meaning where blood lipids were measured, all of them were measured in serum, but in one study that was measured in plasma. The forest plot showed that there was no substantial heterogeneity, and for the geographical location, that's why we created the study source and adjusted for it, to partially account for center-specific effects. And last but not least, the conclusions, it's that consumptions of fats in place of carbohydrates were beneficial only when fats were consumed as MUFAs or PUFAs, that our findings support the global dietary guidelines about detrimental effects of saturated fatty acids, but we disagree with the detrimental effects of total fats, and agree with recent studies that they show that fats are not associated with detrimental effects upon the lipoprotein profiles. Our findings using this new biomarkers, the non-HDL, were in confirmation. And our example, our study shows that federated analysis on diet biomarker associations is possible following FAIR principles and using DataSHIELD. And last but not least, I would like to acknowledge, well, first for our funding bodies, No Money, No Research, and every single co-authored, every single participating partner, particularly to Katarina and Soviets from ENPADASI, my bosses, actually, and colleagues, the project coordinator of ENPADASI, Yilda Bowman, and the dream team, Paul and Dimitris from Newcastle University, who made it possible, the use of DataSHIELD. Thank you very much.
Thanks, Mariona, for this great work. And I want to personally to say a big thank to you and ENPADASI, because it was one of the first projects who used DataSHIELD. And through the collaboration and the feedback we get from you, you help us to improve and enhance DataSHIELD. So thanks for that. So we have a few minutes for questions. Becca?
Yeah, I wanted to just reiterate what Dimitri's just said, which was that I think ENPADASI approached us, it must have been, I think, around 2014 originally, or maybe even 2015, to about us supporting them in using DataSHIELD. And so it's really lovely to see the impact of the project and it being used in your research, and seeing those, some of these final results coming out of that project has been really fascinating. I just wanted to pick up on, there was something you mentioned about, obviously when you first started the project, you might have been using maybe DataSHIELD version 4, and then your final analysis was done in DataSHIELD version 5, and we're now on DataSHIELD version 6. And it is just a comment really rather than a question, but I think that all analysts and users of DataSHIELD need to kind of consider which version they're using, and when they're publishing their papers, I think maybe we would encourage people to mention the version number, because I think later on, if you ever wanted to reproduce the analysis, or I don't know, do something similar with similar data, you know, some of the functions have changed, or over time they get deprecated and they're no longer in the packages, and so on and so forth. And so I think it's useful to know this, I think, as the developers.
Yes, sure. I mean, actually, that you mentioned, we started, I don't know whether it was version 4, but I really do remember that I had, well, the honor actually to test some beta, you know, the beta, like, you know, that .o. So it's like, so basically we had to like run and run over and over again, you know, but it was exciting, the process, you know, I like new things. And I know that using maybe, the classical way of like, yes, having all the studies into a data warehouse and using other SAS, it would have been more quickly, no? But I think it's interesting, that we have to be patient and excited, like to help this project develop, like DataSHIELD, and make it possible for future projects.
Yeah, I think sorry, just last minute, and then I'm going to go. It was just that, yeah, you made reference to the fact that you guys really were using you were testing the functions for us. So this was before they were released in the main branch for everyone else to use. Your consortium was actually testing them for us, and that is what is needed in order to identify potential, you know, risks or issues in the software, or errors, for example, as well. Paul is just saying in the chat that he's echoing the same comments, and congratulations and thanks to Mariona and also an ENPADASI.
And congratulations to you. I mean, I had fun really, because, you know, so for me it was a pleasure to test and, you know, new things. And I'm happy that, you know, DataSHIELD has grown and keeps growing and growing and growing.
Great. Thanks, Mariona. And we look forward for your presentation tomorrow, where you will present the new projects.
Yeah, just a couple of slides, right? So I just made a couple of slides. So, yeah.
There are no more questions, so we can move to the next speaker, which is Stef van Buuren for University of Utrecht, who is going to speak about the strategies in multiple imputation for distributed data. Stef, can you please share your screen?
Yes, thank you. I will make my screen a little bit bigger. So does this work?
Yes, great, thanks.
Okay, thank you. And thank you, Mariona, for announcing this talk and raising expectations. I fear I have to disappoint you a little, because what we're doing is still work in progress. My name is Stef van Buuren. I come from the ReCap project, so that's where my experience with DataSHIELD comes from. When the project started, we didn't know what to do, so we investigated various possibilities. And I remember one other software called Vipar, but we rejected that in favor of DataSHIELD. And what I wanted to do within the ReCap project is to help harmonization using multiple imputation. So once we had settled on DataSHIELD, I needed a package to do the multiple imputation in a distributed context. And so this is about my experiences with trying to implement that. So why multiple imputation? More in general, we always have missing data in our data sets, of course, and it's now a fairly well established technique in many fields. When I switch to the next slide, you see that this is the number of citations. For example, this time series, the big one which you see over here, It's where the word multiple imputation is in the abstract if you search for Scopus. And that one is rising. It's now on at about 500 to 600 citations a year. So, and that keeps on rising. And because, so this is almost linear, this trend, and this is a log. So this is exponential growth. So at some point it will taper off, but still, this is growing very fast still. What multiple imputation does is, if you have missing data, it fills in not only one value, but a couple of values. So that makes it multiple imputation. And under fairly broad conditions, so why do we do it? It provides unbiased estimates of the thing that you want and the correct confidence intervals. And so you can do a lot of things with the missing data, but a procedure that gives you unbiased estimates and correct confidence intervals that's good to have. And that's why multiple imputation exists. So it's broadly applicable. So the impetus was using it for harmonization problems. So if you have one variable with 4 categories, another one with 3 categories, and they are almost the same thing, you can downcode it to 3 or upcode it into 4. But you need to know how the categories relate to each other, and multiple imputation is one way of doing that. So that was the idea of imputing combined data sets. So I've already showed this figure. So let's look a little about how mice, that's software that we use for it, and we use it, I use it because I've written this, this software. So let's look at the complete workflow for if you do a multiple imputation analysis. So we start at the left with a data frame, our data frame with holes in it, so there is missing data there. So that's called the incomplete data. Then we fill it up, not 1 time, but in this picture 3 times. And the imputed data, of course, at the where data are observed, they are just the same, are copies from each other. But where we have missing values, there could be differences. And so you could view it as we're drawing each hole in the data matrix. We replace it by a distribution, and then we're drawing from distribution. And this distribution is very particular to the cell that we're imputing. So I will tell you a little bit more about how we generate those imputations before going to DataSHIELD. Then, so we have 3 completed data sets. Then we analyze each of these data sets by our favorite analysis method. So to estimate the things that we want to know, to do the science, and so on and so on. And so we get 3 results, but we can publish 3 results. We want to have 1 result. So we need to bring together those 3 by averaging, by taking the average. So that's simple to do. And for the variance is a little bit more complicated, but basically it's adding up 2 sources of variance. And at the end, we have then both the estimate and an estimate of the standard error of the estimate. So that's what it does. The mice package implements this sequence of steps, this workflow of going from a data frame to a MIDS object, which is a multiply imputed data set. Once this multiply imputed data set, you can feed it into the with function together with your analysis statement, which is evaluated on each of the completed data set. You get the results. The results are stored in a mirror object, and the mirror object feeds into the pool function, and you get the final result, which is also stored in the pool object. Now, how to get this into DataSHIELD? So that's somewhat of a challenge because it's not a simple one procedure to apply in a distributed way, but it's a little bit broader. And the most difficult step is here, so to generate the imputations. And I will show you a little of what we do if we have just one data set. Everything is in one data set. And what we can do if the data set is partitioned into specific in different parts, and what things we need to be aware of. So first, let's look at a simple scenario where everything is stored in one data set. Suppose that the missing data occur all over the place. What we do then, we specify a model for the first variable, conditional on the other variables, for the second variables, conditional on the other variables, and so on. So this is our imputation model, which is actually a collection of univariate models. And then we simply iterate over those models, creating imputation. So basically what we do, we fit the model on the rows for which we have an outcome, and we apply the model for the rows for which we don't have an outcome, and we add our appropriate noise. So that's what happens. So this is an iterative procedure to create imputations. So we specify an imputation model for each incomplete variable, which gives you tremendous flexibility in what you do for a particular variable. And the whole thing is iterative. So if we make a kind of illustration, so the next is an illustration. Blue is observed and gray is missing. Now we start filling up, the algorithm starts filling up the missing variables by a draw from the variable itself. So here we have 1, 2, 3, 4, 5, 6 variable observed values for variable 1. And what we do, we draw 2 values from those 6 and we fill it up over here. And we do the same for this variable and for that variable. So that's our starting imputation. And then we calculate the imputations for those 2 cells below here, given the values that we have in variable 2 and 3. So, and once we have fitted the model on the first 6 cases, we can apply it to the last 2 cases, and there is our imputation. And we can repeat, of course, this game for column 2 and column 3 and so on. And now we say we've done 1 iteration through the data, and we can start the process again. And we do this 5 to 10 times. It's usually enough to get convergence. And that's our first imputed data set. And the second one is simply it's the same process, but from a different random start. So that's how it works if you have everything in one data set. Now suppose that we have our data set distributed over two locations. For example, within a DataSHIELD setup. The first that we may consider, we could use something like local models. So how would that work? So here the first 4 rows are stored in the dataset number 1, and the last 4 rows are stored in dataset number 2. So these are our distributed datasets. Now, if you look at the first 4, we see that here everything is complete and here everything is missing. So actually mice can't do a lot about it, because we have 2 complete variables and we have no information to impute number 3. For node 2, we can do something. So we can I can start with a draw from those 2 cells and impute it over here. And then do the same game, but on a smaller scale, same game as before. So we fill up column 2, 3, and so on, and we're done. Now, if we summarize this procedure, then we would say, well, what we can do is run mice in each node. So we have different nodes, and we have run mice in each node. And what we can impute is those variables that are partially observed, for which we have blue. And I think what we should do is store the imputations locally, so that, because logically the imputations belong to the data and less to the analysis, although you can have different views on that. So this is relatively easy and fast to do, but it's and it's independent from each other. So each node can do its own process, but there is no learning across nodes. So you don't get imputes for this white column because there was no way that a mice would work on that. And what's also the case is this solution is different from the standard case where we would have a virtual data set with all 8 rows. So this is a different solution. It makes different assumptions. So the results will be different and not, as Paul said in his introduction speech, that the claim statements present the same whether you distribute it or not. And that's not the case over here. So what can we then do if we try to have a global model? So that's the more encompassing model. So let's look at what we can do then over here. Again, we have partitioned it into data sets, and now, for example, to get an initial impute for those 4 cells, we need to draw from all the observed data. So we ask a frequency table from all our nodes, and then draw one of those frequency table and put it in the first cell and that in the second cell. So that's relatively easy to do, and we can do that for all 3 sets. And the next is then, so we can fill it up and make it complete entirely. And then we can start with column 1. And so then we should make a model that encompasses 4 cases from dataset 1 and 2 cases from dataset 2. And well, there's a function in DataSHIELD to do that, and then apply the model to impute those 2 cells. And we can then repeat the sequence for these and these columns. And at the end, we get a fully imputed data set. So that's a little bit of a different scenario. So what we do then is we run mice more at the client level. We sample the initial imputes across the nodes, store the imputations locally, and then per variables fit a model on the blue outcomes and apply it to the red outcomes to produce imputations. And we need to add noise in order to get to account for the uncertainty of our imputes, store the imputes locally, and then the great thing about this algorithm is that it can learn across nodes. So, for example, in dataset 1, there was no variable 3. Either all person, it was missing for all persons, or it wasn't collected, which is more probably the case why it was missing. But now we have a value there. So we enrich, so to say, the node 1 with information from node 2 in this case. So we're really adding value to it. Moreover, if we do it in this way, then it is a standard solution. So it's the same as we would get if we would have access to the complete data set in one time. The big downside is that it has a substantial overhead if you apply this in a DataSHIELD context. And I've made a calculation. So let me first summarize those two, the local models and the global model. So the local model runs at each node, imputes and diagnoses, analyzes and pools, and runs, and the client only combines parameters from the complete data analysis. There's low overhead, and it uses the MIDS object instead of the data. So that's what you need to change then at the local node. And you could say it's somewhat of an advantage that the nodes can do it independently of each other. But the downside is that you get no cross-mode learning in this way of working. In the global model, you can only then you would run mice at the client level, and you would put to work, you put the nodes to work for you in an iterative fashion. And if you would do that, then this would create overhead, and you would need to solve special problems. For example, if you would do predictive mean matching, which is my favorite imputation method, with samples from the data, you have to get the samples from the different nodes, and then find out which ones of all those samples match the best and use that as the imputation. And you need to do it on a per case basis. So that's, and I've made a calculation. If you would have a data set of, I don't know whether it is actual anymore, that imputes 30 variables, and we create 5 imputed data sets in 10 iterations. This costs about 1 minute to complete in R. In the federated context, we need to do a lot of calls. I calculated that 18,000 was my estimate. I don't know, but it's a lot. It's a lot. And when I tried this out, I saw that it took approximately a couple of seconds before I got a response from the other nodes. And you need to wait for the last one in order to go on with the next. So it's the slowest who determines the total time taken. And so for 18,000, it would take 15 hours. And then I thought, well, 1 minute to 15 hours, that's a big difference. Now this may not be actual anymore, but at that time, this was a little bit of a showstopper for me. So, well, I can create this global model, which is ideally, which would be ideal. But if I need to wait for hours to do even a small problem, then that's not really attractive for a user. Also, we hadn't planned for doing this. So recap is about recap wasn't about this problem only, and we had no budget to do this also. So that wasn't more of a simpler explanation why we paused the work. And but I think it's possible to do it, and perhaps things are much faster now and possible to do. We have made a report on this work, which is joint work with Paula Reise and Rui Camacho from Porto in this deliverable, which is now made public, so you can see more in detail what we have done. I think a couple of months ago, I saw an article which dived into the same problem, multiple imputation for distributed health data. And of course, they found the same, that it takes a lot of time to do it to do the global model. They use a little bit of different names. So their local model is called IMI, and their global model is called CIMI. These authors presented simulations about the statistical quality. So they looked at bias and a couple of other things. And this column is the number of communications needed. So you see that the local model is really fast because there's hardly any communication needed. And the full model, the global model, has the most communications. Now, if you just look at the statistical properties, the global model has the best properties, and then it's a standard mice algorithm. And if you start making shortcuts within that algorithm, it will introduce bias and probably also get you lower coverages of the confidence intervals. The authors didn't look at that, but that's what I expect to happen. So that's an argument for using the global, but costs more time. In the, let me see. If you look more at the computational efficiency of those two possibilities, then no communications are needed in their implementation of Y mice, and 25,000 were needed for that problem, using it in a distributed way. So it's a two-sided coin because this takes very long to fit them. See, so where are we now? I think it's fair to say that there is currently no working general package for distributed machine data imputation. You can look into the GitHub repository of these authors from which I just showed the paper. That's mainly suitable to have an illustration of how you can do it for one particular problem, but you need to adapt then the code for your own problem, so it's not generic. So we did work with our Porto colleagues on a DS mice client, but this work it's I would say it's in progress. But if you look at the dates of the last commit, it's about 2 years ago. So there's not really much progress last year. Still, I think it's useful to do. So I would be very interested in if people want to pick up on this idea. And perhaps something in between the local and global form could be that try to solve the problem in 2 steps. So first impute all nodes locally with the partially observed data. And what is left then are the variables that are not observed in data sets. So you fix those local imputations, and once you fix them, then you do the iterative thing, but you need many fewer iterations in order to achieve convergence in that case, because you only need to fill up the variables that were not observed. And sometimes you're not interested in those, and then you can skip it entirely. So this is more or less the update I can give you on the work. Thank you for your attention. I'll be interested to take questions.
Thanks, Stef. I want to say that we have a lot of requests in DataSHIELD for multiple imputations, because in more cohorts, they have a lot of missing data. So I think it's worth on if you can invest some time or to complete your development. And we are happy to work with you if you are interested. I want to say, I don't know if there are any questions, but I want to ask, maybe in a future projects, can the multiple imputations be a part of the harmonization before, for example, if we cannot overcome the challenges about the time needed, I want to say that the users don't have to do the imputations by themselves, every user to do it. Maybe it can be a step before the analysis, and all the studies can impute data, and then they can offer access to the users to the initial data and complete data, and also to the complete data. I don't know if this can be happening in future projects.
Oh yeah, that's essentially the local scenario where each node creates their own imputed data set and then offers that to users.
Yeah, I was thinking also then global can be done through DataSHIELD, but only once at the initial stage of a project.
Yes, that is a sensible scenario to do it in that way, and that's easier to realize than the global one. The only problem is that it's not that it's local, so it's not. You do not get the statistical properties that you would get if you had all the data combined in one file. So that's yes.
Yeah.
I wonder, I think if I understood what Dimitris was saying, was to perhaps take advantage of the way that DataSHIELD now is able to store workspaces so that you can come back to them, so that it might be possible to run the global imputation. Admittedly, it takes a very long time, but then multiple researchers could use that separately for their analyses afterwards. So you run this huge thing that takes a really long time once, and then you have the available globally imputed data sets afterwards. Is that what you were trying to suggest, Dimitris?
Yeah, exactly. Thanks.
So I think, all I was really raising my hand to say was, thank you, Stef. I know you've done a huge amount of work on this, and it's really great to see it presented for everybody to understand what the complications are, because I didn't have a full understanding myself previously. And I think I know Dimitris is interested in working on these issues. I would be interested in working on these issues, and I'm sure there are other people, because I think imputation is such an important aspect of these kind of longitudinal cohort studies that we've been working on, that we've seen some of the results from life cycle and ENPADASI and so on today, and missing data is an issue for everybody. So if we can collectively bash our heads together and think about it, even if it takes another 5 years or something, it would be time well worth investing, I think. So that was all.
I agree.
But yeah, thank you very much.
We have also a question on the chat. Is the problem of the global model specific to PMM, or is it the same with NG and normal imputation model?
There's a little difference between predictive mean matching and normal imputation. With normal imputation, it's enough to know the parameters from the regression model. So if you know the betas and the sigma, then it's enough to do imputation. If you do predictive mean matching, you need on top of that the values of the donors. So then there need to be transfer of individual data values from one node to the other. I think it's not disclosive, but it's an aspect that we should then pay attention to. So there's the difference between the two.
Yeah. Also, we have a big thank from Paul. He has a problem with his microphone. Yeah, and Becca suggests to open a thread in our forum about this. Yeah, if there are no other questions, we can thank our presenters and move to the next.
Yeah, so I will do a little quick wrap up, and I just want to echo what Dimitris just said. Basically, was thank you to everybody who's spoken today. It's been a fascinating day. We've had a huge variety, a kind of huge journey across the DataSHIELD community and the issues that come up. So the ethico-legal issues that we heard about this morning from Meta Hartlev, the introduction to DataSHIELD that Paul gave us, some of the exciting stuff that is being done, for example, by Tanya, using the well-thought developments for the interactions with the small and medium enterprises and to share the data in a different way, the challenges of commercialization. And then this afternoon, a fantastic kind of breadth of real science that's finally coming out of DataSHIELD, or is now really coming out with the projects that were presented by the three different presenters, Johan, Gabriella, and Mariona. So thank you very much. That's really wonderful to see these things coming to fruition, essentially, after these many years. And then, of course, the technical sides of things that have been presented by Sido and by Stef in this last talk. So it's been a really wonderful day. Thank you, everybody, for your participation and for coming. We have another full day tomorrow, excuse me, with a whole bunch of talks. I'm not going to go through them all, but you can see them on the program, on the agenda for tomorrow. I would encourage you again to come and participate, and also to participate in the breakout rooms and just have little conversations with people and do a little bit of networking. I know the format of a Zoom conference is difficult, and hopefully we won't have to do it for that much longer. And maybe next year we can even meet in person. And so just thank you very much. The final little reminder is that there's a small social event that's about to take place in a couple of minutes. Elaine has posted a link in the chat for the scavenger hunt. And so we hope to see many of you there as well. And thank you very much for your participation. Look forward to seeing you all again tomorrow.
Thank you, Andre. Yeah, I think we're going to take a short comfort break, and then we'll be back in 5 minutes. If you feel free to go and grab a drink of your choice, maybe a cup of coffee. And so, yeah, we'll start the social in 5 minutes. Thanks.